All right, thank you. I don't know about you, but my brain is kaput. Hopefully, you'll still be able to follow me. Thank you. So, yeah, Florida. So every time I said that I'm in University of Florida, is that the pointer? Yeah. So there's people, I see the mental image picture in their brain, something like that. Or depending on your gender preference, something like that. But I assure you, there's people working, doing science in Florida, not only drinking mojitos and spending time at the beach. For example, there's people here working. Not me, but these two. But the environment is very different, actually. In Florida, we're talking about environment and the host and so on. I mean, if you buy and shop a Christmas tree in Florida, you know, the dress code is very important. Sandals, this is the family here. So sandals, shorts, top down, this is the Christmas tree here. Palm trees around, you stop by because it's so hot, you eat an ice cream. So this is a different environment. So it's very difficult to do science there. I don't have postdoc position open, but please contact me if you think that it's a fit for you. So at this point, it's kind of redundant because you get the message that there's microbes and they live around an organ that could be susceptible to cancer. And we understand the phases, the cellular progression of the transformation of the tissue. And we have a good understanding of the molecular transformation mechanism that the cells go through. And that's that. Well, the thing is, in this organ, there is, as we learn, I think if you were paying attention, there's trillions of bacteria. And what do they do there? Are they a bystander or are they active participant? In the plus or minus, so refraining the cancer progression or enhancing cancer. And this is the big question that I think this session has been addressed, as trying to address for a long time. And we know there is differences in the microbiome of case control. The question is, what's the functionality of that? And this is a bit what we do in the lab. So we know that the cancer has a strong genetic component. There is dietary factor. Inflammation, I'll come back to that, is a very powerful environment that shape the tissue, but as well as the microbes. That's the topic of my talk. There's an increased rate of cancer, colorectal cancer, in people that have chronic IBD, so it's a very important factor. It's a small segment of colorectal cancer, but still very significant. And do we add the microbiota here in this equation? Yes or no? And I think we're leaning toward yes, but we need to understand more what is the relationship, causal relationship. And if you think about that, this is your gut when it's very healthy after a prep. So nice and shiny bowel. And this is the progression when you have disease, mild, severe colitis. And you think about that now, there's a lot of bugs in this environment. Now that has been cleaned, but typically there's a lot of bugs. And they interact with this tissue. And what's the outcome? You know, what is this relationship between that was a normal, very healthy barrier function intact, and this over the course of decades? So it makes sense to study inflammation in the context of microbes' activities. And we published that paper. I'm just going to use this cartoon to sum up because it's been a year and a half ago. But that was our question. Do you have a different microbe? This is the mouse, a mouse model here. So what we have is that the microbes are important to initiate colitis. That was a susceptible murine model. So they were susceptible to disease. And you introduce microbes, you have this strong inflammatory reaction in the environment, the tissue environment here. But this inflammation was also impacting on the microbes' activity, and they turned them into what we call a genotoxic microbes constituent. So, and an example of what we found is that there were a specific gene associated with a bug that we all have, Escherichia coli. Here, the enemy is within. I'm not talking about an infectious microorganism that is entering the system. I'm talking about something you carry over, but for some reason, either lifestyle, diet, and so on, they change their behavior. And this is very fascinating to know that you may carry something that is healthy, but could turn into the pathogenic aspect depending on the cues you receive. So here, the gene that we had was called polyketide synthase. So it's a natural product produced by these E. coli. Again, it's a commensal E. coli. It's not an infectious microorganism. And this gene is very complex. There's 22. It's a polyketide, so it's not ribosomal straight to peptide. It's multiple enzymatic activities encoded by different genes. And these activities shape and build this product that we have not purified, not characterized. So it's a lot of work to do. And what we did, basically, we took this E. coli and deleted that gene. So we moved the genes out. This is a COMET assay. This product, polyketide synthase, PKS, is doing double-strand break DNA and then force repair mechanism. If you don't repair correctly, you carry mutation. So this is an example of a cell incubated with the bug, and you have the COMET tail, which indicate DNA break. And when you remove the PKS, you don't have this phenotype. And we went to the mouse, and this is a monoassociated system. So the mouse is called an IL-10 knockout. You introduce the bug, so it's a one-bug environment. And we look at inflammation. This is an inflammatory score. So the NC101 is the name of the bacteria, and the deleted hydrogenic strain. So inflammation-wise, if you introduce the wild type and the mutant, there's no differences in the inflammatory reaction of the host. So that compound or that drug or that product doesn't shape the host's inflammatory response. So that was what? That's interesting. But we look at cancer then, and what we saw with the cancer, which is here invasiveness or tumor multiplicity, then we saw a difference. And that's quite fascinating because there's a lot of, the concept and the dogma is that chronic inflammation will push you to cancer. Now I'll just show you this one slide before. That if you delete that gene, you don't impact inflammation at all. The inflammation is there, but there's no cancer. So which makes you think that inflammation is not only by itself sufficient to shape to chronic, to cancer, but you need microbial activities. I'm not trying to explain that colorectal cancer is due to these genes. I'm just saying it's a proof of principle that it could. But we were interested in that gene because there's a lot of E. coli isolate that were found in colorectal patient and also IBD patient. They are there on the mucosal. So we collected from collaborators these E. coli and look for the gene. And what we found is there is almost a threefold increase from the control. This is non-cancer, no IBD, compared to colorectal cancer. So there's a higher prevalence of this PKS gene in human E. coli. So that was interesting. And that was duplicated by an independent group. So these people are in France, I think. And they basically found the same kind of ratio. This is in percent, almost 20% baseline and 55% increased prevalence of that gene. So this is the independent cohort, so we felt very good about that. So how do you get there? So I just told you that inflammation, non-inflamed and inflamed, you have different bios, different microbial community. Oh wow, that felt good. So how do you study that? You can do prospective studies in human. So case control in human, I think we understand there's differences in microbiome. We are in dispute over who is more abundant or less abundant. We're still trying to settle this down. And sometimes you may not need it. If you need a biomarker, you know, that's one thing. But if you try to explain the disease caused by one microorganism, it's an issue. But let's do the mouse, because the mouse, you could manipulate that. So this is a biobubble, the germ-free isolator. And you have your control mouse, this is a normal mouse, and the IL-10 susceptible mouse. So there's no bacteria in that setting. So what you could do, you move these mice out of the bubble, and the cohort, you collect stools over time. So you follow microbial composition assembly over time between a non-inflamed and a chronic inflamed cancer mouse. And we did V6 HiSeq 2000 sequence. And the biobubble I'm talking about is that this is what we have in Florida. So the mouse are kept in germ-free isolators. They are healthy, happy. Once you move them out, then you will settle or start the inflammatory response. So this is something that you've seen before. So this is a wild-type mouse, this is the IL-10 knockout. So normal, healthy, colitis, cancer. And all these symbols of the microbiome, the entire taxa assembly of an individual mouse. So all of these are in single mouse. So this is 2 weeks and the IL-10 knockout here. And then you follow the same cohort over time because the biome will assemble, will mature, because that's the way we assemble biota. You're not born with one. You know, you take times to assemble. So what we did here, we follow them, and you could see at 12 and 20 weeks, there's absolutely almost no differences. So the maturity of this biome is almost reached at 12 weeks, and it doesn't really matter if you sample at 20 weeks. It's basically the same, and the metrics are there. But what happens if you have a mouse that progressively develops colitis? And you have a different pictures beside this outliner here, the cluster in that region. This is 12 and this is 20 weeks. So the assembly is not like that. They don't mature in one spot. They kind of step with colitis, high colitis, and colitis cancer. So that was an indication, not a proof, an indication that inflammation do influence how you assemble your biome. Now you could drill down in this individual mouse, who is the contributing factor or who is contributing to the differences. And when we did that, we saw in the family of Enterobacteriaceae an increase in the IL-10. This is the 2 weeks, 12, 20 weeks. There's always an increase of Enterobacteriaceae, which is the family for E. coli, over time. So, but they are select out. The E. coli is usually select out. It's a very low abundance bug. But when you have inflammation, the niche resists better. They occupy that niche with a higher prevalence than without inflammation. And if you go, because this is high-seq short reads, we don't go to species and genus, but we were able to match this to Escherichia and Shigella, and it's the same pattern. So you enrich with inflammation that niche. And that was interesting for us. So we said, okay, so if you have cancer, you say, I have a high abundance of fusobacteria in that case. And I'm going to mimic that, or you have an increased abundance of E. coli cancer. I'm going to go and put a load of these bugs into a murine model. And I'm going to ask the question, do I have more cancer? There's no competition there. You know, you just come, artificially nourish the mouse. And that's what we did here. Because in a germ-free, if you come in a monoassociation fashion, there's no competition to set the niche. Because there's nothing to fight. So the bug is coming and colonize this mouse. And that mouse here, we tricked it by removing the RAG2, which is, and you learned that before by Matthias. So there's no mature T and B cells, so, which is the engine of chronic inflammation in that model. So basically that mouse will have the same IL-10 deletion, but that one can't progress to inflammation. That was the thought. And then you associate with the bug I just told you cause colitis cancer. And then what's the outcome? So predictably, if you don't have a T and B cell, these mice don't develop inflammation, and the mouse that have IL-10 single knockout do have inflammation. So we say, okay, no inflammation, what happened? Remember, this is highly loaded for a bug that induces cancer. And if you look at cancer, you don't have the development of cancer in that model. The IL-10 knockout alone will show cancer, but not the double knockout. So you need inflammation. And this is not because, that's the histology, this is not because you're lacking E. coli, this is the CFU load in these mice. So, you know, the one that doesn't develop cancer has as much E. coli as the one that do develop cancer. So these bugs are there, they're just dormant. So they need a cue. Which kind of cue do they need? Because load of bacteria in that case is not the answer. So if you increase abundance in this model, you don't get cancer. So there's something else missing, and there's something else that may be provided by inflammation. So it's a very complex interaction. Inflammation hit the host and also hit the microbial activity, well, assembly and activity. So how do you answer that question? We did another simple experiment, again, biobubble germ-free, the wild-type mouse, the IL-10 that doesn't have inflammation, monoassociated over time, But this time what we did, we isolated microbial RNA to do RNA sequence. So the question is, are the genes that are involved or modulated by inflammation? And how are these genes related to cancer? Because the hypothesis was that the inflammation is a shaper of microbial activity. That was an interesting experiment. Cost money. So this is what we found. And it's almost the last slide, so bear with me. So what we have here is 2 weeks, the same scheme I just showed you, 2 weeks, 20 weeks, 12 and 20 weeks over time. This is a transcriptome assembly of the E. coli for that 2 weeks, 12 weeks, and 20 weeks. So we said, it's fantastic. The transcriptome of E. coli change and polarize and cluster over time because of inflammation. The thing, though, is that you have to do control. And the control is this. The control is the mouse that doesn't have inflammation, right? And they almost superimpose to each other. So if you remove inflammation, what you see at the end of the day is a change, adaptation of the bug to the environment. Maybe 0.3% of the entire genome of E. coli is responsive to inflammation. So we were wrong in thinking that that was a massive bomb that would change the transcriptome. It's not true. It's a very small fraction. Well, that's an interesting answer by itself. A lot of the genes that were changed were adaptation genes, and that's beside the point. I told you about the PKS, and I'm getting to almost the last slide here. So this is how it works here. Those are contigs, because we haven't fully sequenced the NC101 genome. So those are contigs which carry a lot of genes, so operons. So this inside are contigs, this are operons, and the outliner are the genes under the operon. So that's 2, 12, and 20 weeks. You see that the money shot is here on the p-value. There's a lot of genes changing here. The size of the node is the size of the p-value. So the bigger the node, the more changes, significant changes, so the more swing you have. And so there's a couple of things that pop up. So this one here, those where we're looking at them, and they're all ribosomes, mostly housekeeping genes. So it's interesting to see them showing up as strong. We don't know the meaning of that in terms of host response, but that's what we see. The next one on the list is this guy. So remember, that will be this one here at 2 weeks. And then that one, the same node at 20 weeks. So there's no changes here, and it's increasing. And this one is PKS island. So there's a couple of genes, 5 of the PKS island genes that highly increase during development of inflammation cancer at 12 weeks. So it's a responsive island that is transcriptionally modulated. We try to understand the role and function of these genes in terms of inflammation cancer, but one will predict that it's very important to modulate the cancer impact on the host. And yeah, so everything I've shown you so far is mouse. Well, why am I sitting here listening to you? And I'm almost done. So what we wanted to do, I told you the gene we found it in E. coli of human, but we're interested to know, E. coli is one of the first bugs that is colonizing a human gut, the infant. So we went to publish data, and you can't read that here. Those are population for necrotizing enterocolitis, so a disorder of the gut in infants is deadly. So we were able to crunch sequences, and the most abundant differences between the neck and the healthy is Escherichia and Klebsiella. Klebsiella is a PKS positive strain. Only B2, E. coli, and some outside bugs like Klebsiella have PKS. So we contacted these people because I knew they were in UF, and went to the stool of these infants, and we found PKS positive in the stool of these kids, and we got isolate, a Klebsiella isolate from an infant, and tested it in this COMET assay. You could see they are genotoxic. So the question is, if you have a genotoxic bug very early on in your gut, what's the long-term consequences? And could you manipulate the genotoxicity here by having a good lifestyle? And something could dysregulate the activity of these bugs and the abundance. And that associated with cancer. It's an open question, interesting hypothesis, and we're working on that. And that's over. So what I wanted to say, and I think a lot of people have presented that, there is a dysbiosis, maybe some bugs are associated with cancer. What could you do with that? You try to manipulate, their identity, remove the bad guys, come with new ones. Some people will believe that probiotics will get you there. I'm not quite sure about that. Bacterial therapy could be. It's been done and currently done for C. diff. How about you have a microbe that, by biomarker, like Pierre is working on, said, yeah, you have a chance of developing cancer. Let's remove these bugs, and I give you a brand new set of bugs. So that could be. And obviously, a dietary manipulation of activities make them more butyrate producer for cancer, probably. We're targeting already some genes. I mean, the PKS gene I told you, there is coming a small molecule inhibitor coming for that. It's not crazy to think there will be other inhibitor of different function that associate with cancer. And the same for inflammatory genes. But the idea is to prevent this dysbiosis and maintain a normal colon. And thank you for your attention, and hopefully we could be healthy when we start eating. Thank you.
Thank you very much. Are there questions? Are you all hungry?
Yeah.
But nevertheless, I would like to ask one question. This PKS gene, I mean, you showed abundance of the genes, of the gene in different populations, colorectal cancer, IBD patients. So what's the regulation of the gene? I mean, one is presence, the other one is the capability of modulation.
Right, so that's the question we try to answer. No one knows the regulation. People have done in vitro manipulation of the gene. It's very resilient to cues. So that is the first time in vivo that we could see swing of the gene, of some of the genes. There's 22 genes. But we don't know the impact because you can't measure the product and can't measure, you could measure the activity, is difficult. So at the end of the day, it's just an example of a gene that could be manipulated, but the functional consequences, we're not sure. We have to delete some of them and study that.
One last question, and then we are done. We're still 5 minutes better than the other session, so it's okay.
On the very same night, I mean, that's an antibiotics resistance gene, so it goes up for all kinds of chemical treatment. It could sort of go up.
You say antibiotic resistance?
PKF, I thought, yeah. So it's a polypeptide synthesis?
No, it's not an antibiotic.
I thought it was an antibiotics resistance producer.
No, we don't know. I mean, I wish. If you have that, we'll publish together in Science. No, we don't know what it is. I mean, polypeptide.
Okay, so you only have
right, yeah. You're right, in polypeptide, penicillin, and all of them natural product are antibiotics. This one, I have no clue. But, yeah.
Yeah, so because treatments, those genes are most flexible for all kinds of treatments.
So the bug is likely to maintain, this is a big 54 kb, so you have to have a reason why you carry it. I'm not going to tell you the data we have, but I mean the idea is beneficial for the bug. Either it's killing other bug, but it doesn't do it like a typical antibiotics. But it's a work in progress.
So thank you very much.